On behalf of PHA Canada, welcome to our 2025 webinar on insights from recent clinical trials in pulmonary hypertension. I'd like to introduce Dr. David Langloben, our presenter today. Dr. Langloben is a professor at the McGill University Faculty of Medicine and former chief of cardiology of the Jewish General Hospital. He's the founding and ongoing director of the Centre for Pulmonary Vascular Disease, the first pulmonary hypertension clinic in Canada. His research interests include lung vascular metabolism, the behavior and function of lung vascular cells, vascular cell biology, the epidemiology of pulmonary hypertension, and the development of new medications for pulmonary hypertension. We're very glad he agreed to put this webinar together for us. So over to you, Dr. Langloben.
Thank you so much, Robin. I guess good afternoon to everyone, and I'm really grateful to PHA Canada for having invited me. Okay, and I'm just going to make do you see the laser pointer? Excellent. Okay, well, welcome. We're going to be talking about insights from recent clinical trials, and I entitled it Progress or Not because we've had successes and failures. And I think it's important to discuss all of them so that people really understand the medication development process and its hurdles in many ways. These are my conflicts of interest, and I am going to be talking about medication today, so I really do want to declare these conflicts of interest. It's more conflicts than conflicts of interest, so just be aware that I do consult to various companies. Okay, to be able to understand the drug development process, we need to understand the various stages of drug development, and there's sort of three key phases that you'll hear about quite quickly, which are phase 1, phase 2, and phase 3. But before that, we need to be able to identify molecules and medications, potential medications, that might have effects. And sometimes that involves massive testing of compounds. I was recently at a pharmaceutical laboratory in Germany, and they literally have a giant warehouse with machine robots putting drops of various chemicals into test tubes with cells growing in them to be able to test tens of thousands of chemicals at the same time and understand their effects on cells. So let's say something looks interesting. At that point, several things may happen. First of all, if it's a medication that is already in use for other purposes, some investigators may choose to do what's called a pilot study, which is say, hey, maybe it's a good idea to try this out in a small single center cohort of patients with pulmonary hypertension, or even in a single case, a single patient. And you're going to see examples of that as I speak today. But more likely, what's going to happen first is that the medication will be tested in humans, normal humans, to see what its side effects are. Safety is paramount to us, even over and above efficacy. We can't use something that's effective if it's not safe. We need to know that things are safe as best as we can tell. Also, the phase 1 studies let you decide, when you take a pill or take a medication, how long does it stay in the body? How is it destroyed or excreted? And we gain early insights into the correct dosing. So after finishing phase 1 studies, it gives a company the basic information to try and go ahead and say, okay, how would we then structure it for a pulmonary hypertension trial? And that involves a phase 2 trial. And a phase 2 trial is actually in patients with pulmonary hypertension. Usually a range of doses are studied because you want to know the lowest effective dose ideally, and also, the highest dose tolerated. It is usually placebo controlled, meaning a group of patients will be receiving nothing or a dummy drug, because results do change with time in patients receiving nothing. The disease will either progress, or you have to be sure you remove psychological effects. So for instance, if I take a sugar pill, I might be able to convince myself that I can walk 20 meters more. And so you need to always compare the medication you're testing versus the placebo. And initially, you want to study what it does to the hemodynamics of pulmonary hypertension, because pulmonary hypertension is, of course, high blood pressure in the lungs. And so the endpoint that's most often used is something called the pulmonary vascular resistance. You're looking at many secondary endpoints. In other words, can they walk more? Is the heart working better? Do the levels of the NT-BNP, which is a hormone that's produced by wall stress in the heart, do the NT-BNP levels come down? You look deeper for side effects and safety. And these studies often provide some proof for approval. And then finally, the medication has to be checked in a large placebo-controlled phase 3 trial, which actually looks at important functional and prognostic endpoints. In other words, um, uh, can they walk more? How's the heart doing? Is it better? Does the NT-BNP come down? And what's the prognosis? How do the patients getting the active drug do versus patients getting placebo over time? Is the quality of life better? Side effects are studied. And when the study finally comes out well, at that time, a company can go to the regulatory agencies, Health Canada, Food and Drug Administration in the States, and say, look, we think this is effective. And those agencies have to give their approval. So first, we need to understand what PAH is. And this is a old slide. As you can see, it's a very old slide, but it suggests that there was, if you have very small pulmonary arteries that are normally wide open, that they gradually start narrowing and get plugged up with cells. And while this schema is incorrect, and I'll explain why it's incorrect, I think it's a nice, pretty picture of what's going on inside the vessel. There's piling up of cells and narrowing of the vascular channel. And you can imagine that if this gets tight enough, it's not going to let blood through. And imagine then that there well, hundreds of millions of these little vessels, and that they're slowly getting plugged up. So what's wrong with this schema? This schema assumed that this process started late, but we had very bright scientists early on, and this is a paper from 1958, and they knew that the blockage, which was cellular, was actually occurring early in the process. And really, it's the fundamental problem in PAH. PAH is a disease where the cells pile up inside vessels, inside the channels. Imagine your bathroom sink drain sort of slowly clogging, okay, until it really becomes very clogged. So over the years, we've learned that we can take some of our medications and try to deal with the various biochemical abnormalities that occur in parallel with that cellular plugging. So we use agents to block a bad molecule called endothelin, and you may know macitentan, you may know ambrisentan. Some people may still be on bosentan. We know that there's not enough nitric oxide, and so we play with the downstream signaling to try and preserve the effects of the nitric oxide. And we do that with things called phosphodiesterase inhibitors, sildenafil and tadalafil, or we can try to crack up this enzyme here called soluble guanylate cyclase with a medication called riociguat. And finally, we know that these abnormal cells don't make enough prostacyclin, so we replace the prostacyclin. And some people may be on intravenous epoprostenol, some may be on subcutaneous treprostinil, or some may be taking an oral medication that interacts with the receptor here, and it's called selexipag. So these are our three arms of therapy, or were our three arms of therapy, even as recently as 2025. And this is a schema. This is from 2022, but until recently, this was our schema. Patients would come along. We'd start them on one or two therapies, sometimes three therapies if they were really sick. We'd follow them. We'd sort of, play and match the various therapies to try to find the best combination. And sometimes when they were sicker, they needed intravenous or subcutaneous prostanoids. And this is really what we've been doing for the last 15 or 20 years. And it has helped tremendously. And patients improve, do better for a while, and respond and have better lives and longer lives. But we also know that we really are not modifying the cell problem in the vessels. And this is someone who was on intravenous epoprostenol for, I think, it was over 15 years. And you can see this is the channel that's left for the blood cells here. The vessel still severely, severely narrowed, which raises the question of, well, this person was doing pretty well functionally. How are we helping them? And it may be that we were helping the less diseased vessels and that this process just continues underneath. It may be that we were helping cardiac function a bit. So all of our therapies do help, but they don't stop the real underlying process, which is the cell growth. And so it may be that our treatments really, while very, very good, and they buy people 5 or 10 years, just aren't up to the disease. And we need other approaches. And we've now switched our thinking. Of course, we're still using the agents that have proven effective over the years. They do offer benefit, and they offer rapid benefit to patients. We're now looking at a variety of agents that affect cell proliferation, that growth and plugging of the cell. And the first group I want to talk about is a group of agents called activin ligand traps. And the one that's sort of farthest in development and really just we're very, very close to having it very widely used, I hope, is an agent called sotatercept. And to understand what sotatercept does, you need to understand what's going wrong. All of the cells in our body have thousands and more different kinds of receptors. It's how cells talk to each other. Molecules float around, and then they signal into the cell by attaching to specific receptors, okay? And receptors can be good, receptors can be bad. So all of our cells in health, when we're healthy, are in balance. There's a group of receptors called bone morphogenic proteins. The bone morphogenic proteins bind into bone morphogenic protein receptors, and those tend to be anti-proliferative. They keep cells, the lung cells from growing, the lung vascular cells, and they maintain a nice, healthy, open channel. On the other hand, there are molecules that are actually, they're very important at other times for cell growth, but in this case, they have a bad effect. And they're called activins, and they bind into activin receptors. Now, normally, this is in balance. The BMPs and the activins are in balance, and the cells are healthy. But if something goes wrong with the bone morphogenic protein side, either there's a mutation in the receptor or a disease affects the amount of receptors being produced, or there are problems with the bone morphogenic protein, you lose this signaling. And the signaling tips in favor of the bad stuff, as you can see here. And when that happens, the cells all proliferate and the vessel gets very sick. And this is what seems to be one of the fundamental, maybe not the most fundamental, but one of the fundamental issues in many types of PAH. Well, what is sotatercept? Sotatercept is a molecule that binds up these activins and keeps them from binding into the receptor. So it doesn't affect the receptor, but there's less signaling from the receptor. And even though this is still sick on this side, it rebalances things and restores vascular health to a great degree. And we now have landmark studies with sotatercept. The STELLAR trial, which compared to placebo, a phase 3 trial, which looked at patients that had been on all those other PAH therapies that I showed you for many years, were stable, but were certainly not well. And you can see that the sotatercept dramatically improved what we call event-free survival. The ZENITH trial looked at our worst patients, the ones that were very sick and on intravenous epoprostenol, but sick on intravenous epoprostenol. And you can see that again, sotatercept had a dramatic effect versus just being on our regular therapies. When this says placebo, it's not that the patients are getting nothing. They're getting everything else we can hammer them with. And Hyperion trial did an interesting thing. It said, okay, new patient, okay, we're going to start them on as many of the other PH therapies as we can. And after several months, we're going to add the sotatercept quite quickly, okay? And you can see that starting the sotatercept already had a dramatic effect early on. We're going to ignore this just in the interest of time. So we have 3 landmark trials, and sotatercept is approved by Health Canada, and the PCPA approved it in principle last week, and now it's up to the provinces to figure out how to pay for it. So we're going to have to see how it evolves, but certainly many patients, not every patient, but many, many patients are going to benefit from this. And these are our most recent guidelines for pulmonary hypertension physicians. And you can see that before, whereas we were talking about all the other agents, yes, we still start with the other agents, but rapidly we decide how our people, and if they're not in low risk, you'll notice that in all instances, there's a suggestion to add an activin signaling inhibitor. And so I think sotatercept and maybe other activin signaling inhibitors will be used widely. So we're going to rate that as a success. I'm going to be rating things as success and failure today. Now, just to give you an example of maybe not a success, this is a study of another activin signaling inhibitor, which should have worked. And this is the TROPOS trial, and the agent was given to patients versus placebo. And there was an unexpected side effect. And the unexpected side effect was that many, many, many of these people developed pericardial effusions at a very high rate. Pericardial effusions are collections of fluid in the bag around the heart. Placebo only, you need placebo because look, even 10% of patients with placebo got pleural pericardial effusions. Pulmonary hypertension alone can give you a pericardial effusion. And that's why you always need a basis for comparison. But still, it was much higher with this therapy. And there are a lot of theories as to why this might have happened, but the study had to be stopped. Now, sotatercept has also been, there were case reports of pericardial effusions with sotatercept, but it's certainly not with this kind of frequency. And the effusions don't seem to be dangerous in terms of compressing the heart. They're just present. And nonetheless, this was such a high rate that the study had to be stopped. And it shows you the perils of the drug development process. This company spent a lot of money on phase 1 trials, spent a lot of money on a phase 2 trial, spent a lot of and got this far, and suddenly there's a bad signal that was unexpected. And to show you how perilous this is a small company that probably depends on investment capital. This blue line is the stock value. And the day they announced that the study failed, basically the company tanked. And so when you see stuff like this, it's not surprising that particularly the small companies are very careful and actually very hesitant sometimes to develop novel medications, which is unfortunate because good ideas have to be tested. You know, if sotatercept was a good idea, if sotatercept had not been tested, if they had said, "Oh my, you know, we can't afford to take the risk of an issue happening," we wouldn't have sotatercept today. And so the way we try to mitigate that risk is by doing very careful trials with very close observation of people. And if we get a signal, we halt the trial. So this trial was halted. It's not like the study was finished. All studies have what's called a data safety monitoring board. And they're an independent group of physicians who are independent of the study, have no conflict with this study, and their only job is to look at the data at regular intervals. And if they feel there's something, a signal of a lack of safety or of a particular complication, they notify the pharmaceutical company and say, "Hey guys, it's time to stop the trial." And that's what happened in this trial. Okay, so no one was damaged by this, but you can see that companies can really go under very quickly. So no success with that. Now, there's another group of agents called tyrosine kinase inhibitors, and tyrosine kinases are enzymes that are associated with receptors that are involved in signaling. Look how complex the downstream signaling is from a single receptor here, okay, for a single molecule. It's such a sophisticated system, and there's so many different places that can go wrong, and there's so many different places you might be able to make it better. But they regulate key cell functions such as cell growth, cell maturation, and what's called anti-apoptotic signal. Apoptosis is programmed cell death, and so if you lose your apoptotic signaling, cells pile up. Okay, so again, it may be contributing to processes. So receptor tyrosine kinases, there are many, many, many of them, And there's whole families of tyrosine kinases, and this graph shows where each of these medications, which tyrosine kinases they affect. And I want to highlight imatinib, which is actually very selective. It doesn't do most of them, and compare it to sunitinib, which hammers basically all receptor tyrosine kinases. All these agents are being used in different types of cancer. And remember, cancer is cell proliferation. So um, uh, one could say, well, maybe the agents that our oncology colleagues are using would be useful in PAH. And that's exactly what happened. And at full credit to Dr. Ardeshir Grofrani from Germany, who had been treating his patient with all the various stuff we use for PAH. A patient wasn't doing well, and he sort of walked down the hall to his oncology colleague and said, "What do you have?" And they had them in this bottle, and they said, "Take 2 in the morning and call us." Well, it was this agent imatinib. Okay, I'll go back for a second. It's this one here. Okay, and he gave imatinib, and lo and behold, in a single patient, um, things got better. Okay, and he published it as a letter in the New England Journal in 2005, 20 years ago, almost 21 years ago. And then there were other case reports. This is what I mean about single center case series. Other case reports suggesting there might be an effect, but it had to go further. And it did go further in a phase 2 trial called the IMPRESS trial, where they gave patients imatinib or placebo. And you can see that the patients with imatinib did better, and that their hemodynamic effect, the effects on the various things we look at, the pulmonary vascular resistance, the pressure, et cetera, they did better with imatinib. And when you look at the various types of other PAH agents they were taking, double or even triple therapy, it offered benefit, right? So looks like a great trial. Why don't we have imatinib? Well, imatinib, and all my oncology colleagues tell me this, it's got a lot of side effects, okay? And people don't like taking it, and people drop out. And in this study, more people in the imatinib arm dropped out. You'll see at the end here, there were only 66 versus 80, okay, despite the fact that a similar number had started. So they dropped out, but they didn't drop out because of pH. They dropped out because of the other side effects. And so, although all these things look the right way, the Food and Drug Administration said, you know what? Too many dropouts. You got to do another trial. And frankly, the patent was ending on imatinib, and the pharmaceutical company that owned it said, you know what, we're not spending another $25 million, which really was our loss, okay, because it probably was a very effective agent, and my colleagues in Germany still do use it. It's just not available to us. So how else to deal with imatinib? Well, imagine you could get imatinib in by breathing it in. And by getting it to the lungs, you use a device that lets you inhale it, and it travels down in the airways, but then it diffuses into the blood vessels that are blocked. And the advantage of that is when you take it by mouth, it goes everywhere in the body. When you take it in the lungs, most of it stays in the lungs. So, great idea. There was the IMPACT trial of inhaled imatinib. And the problem is it failed. Okay. It did not. It was a phase 2 trial, phase 2B. Okay. It was going to go to phase 3, but never even got there. It did not meet the endpoint of change in pulmonary vascular resistance for any of the studied doses. And there's a whole bunch of reasons it could have happened. Maybe it wasn't getting in well enough. Maybe the dosing wasn't right. All these things are guessed at when you do a trial. So when you got a negative study like this, you don't really know in the end, would the stuff have worked or not? But again, this was a small company, and they basically went under as soon as this happened. And so this is not being tested anymore, unfortunately. There will be other studies probably looking at other forms, but I want to tell you, so no success on this one. There is another tyrosine kinase inhibitor being tested, which is called seralutinib, and it touches many of the similar receptors as imatinib. These are the series of receptors, okay? And so it's not imatinib, but it's quite similar, and it's been studied, and it's been studied in phase 2, a phase 2 study, and it seems to have effects that go all the right way. So right now, it's in a phase 3 study, and we should have more news in 2026. So why is it important? I told you before we have sotatercept. Well, it's important because first of all, um, not there's no medication that's going to work for everybody. We need other medications and we need other anti-proliferative medications. But what I'm really interested in seeing also in this phase 3 trial is many of the patients that were given seralutinib were also receiving sotatercept. And um, this offers the ability to begin to look at should we be combining antiproliferative agents. In many cancers, people don't take just one agent. They take agents that attack different pathways as antiproliferatives. And it could be that the future of PAH is a patient taking several antiproliferatives. We'll have to wait and see. So, success so far. Now, what about some other agents? There's a molecule called serotonin, which circulates, and the lung has a lot of serotonin receptors. And it's known, it's certainly in animal models, that serotonin affects cellular proliferation. And we've known this for years and years and years, okay? It affects cellular proliferation, in theory, could contribute to the development of pulmonary arterial hypertension. So, logical to study serotonin. And we've known for years that if you block serotonin receptors, this in this case with an agent called ketanserin, you can get an acute response, but we didn't know really what it would do chronically. There's been a recent study of an agent that reduces levels of serotonin called rodatrestat. It was called the Elevate 2 trial, and you can see again, it was a phase 2 trial of rodatrestat at 2 different doses versus placebo. This is sort of a typical PAH study. And it was logical to try this. And what happened? Well, it goes to show. Here's your placebo, okay? This is the percent change in pulmonary vascular resistance over the study, okay? And you can see that placebo didn't change much. However, the people with rodatrestat got much worse. The pulmonary vascular resistance went up. Luckily, didn't cause major harm at all, okay? But you can see that this medication, which should have helped, didn't. And there was no way of knowing it in advance. If you look at the NT-BNPs, which indicate cardiac stress, the cardiac stress went up in rodatrestat versus placebo. And if you look at the change from baseline in NT-BNP, most people on placebo didn't do much. You know, there were a few people that got worse. It's normal with placebos. But look at this, you know, at either dose, the great majority of people had increases in NT-BNP. So this went absolutely the wrong way. And so it shot the theory down. Okay. And so we think now that maybe serotonin's protective. And it goes to show because this had been tested in animals extensively, and there was no suggestion that serotonin was protective. In fact, blocking serotonin helped animal models of pulmonary hypertension, yet it didn't in humans. So no success. We've known for a long time that female sex is a risk factor for pulmonary arterial hypertension, and that men and women with PAH have higher estradiol levels. Estradiol is one of the female sex hormones versus controls. And here's estradiol, and the enzyme that activates or converts to estradiol is called aromatase. Okay, and aromatase inhibitors are actually used in breast cancer because estrogens, of course, can have detrimental effects on breast cancers. And this should be anastrozole. It's a typo. I apologize. Anastrozole is an aromatase inhibitor. And so this was a very well-designed study, and interestingly, not driven by a drug company. This was just a bunch of scientists getting together and saying, we should test this. And full credit to Stephen Kowit, who's in Philadelphia, because he does these trials and he does them really, I think, on a shoestring relative to sort of what the pharmaceutical companies spend on drug trials. So he did this study, and he showed, in fact, that anastrozole, which is the red here, Okay. The benefit in terms of cardiac effects and NT-BNP was really not that present, although 6-minute walk distance did go up. Okay. And it was sort of mixed signaling, but it wasn't a particularly strong benefit. So no success. But because there's sort of signaling in here, and anastrozole is a relatively benign agent, the question is, okay, do we design it differently and try again? So we're waiting to see what will be developed from this. Now, what about treating early PAH? First of all, it's hard to find early PAH, but when we do, what about treating it early? We know that from, again, a large study recently, if because pH was traditionally defined as a mean pulmonary artery pressure over 25. We know that 20 is the real upper normal. So the question was, okay, what do we do with people who come in with pressures between 21 and 24? And this was sort of a large, dirty study of patients. And what they found, interestingly, was that if you treated patients, it wasn't such a large study, but there are others supporting it. If you treated patients between 21 and 24, their prognosis was better. And this is 5 years since the right heart cath, versus just saying, oh, you know what, it's very mild, we're not going to do anything about it. So this pushes us to try to treat people earlier. Now, what about non-group 1 PH? You know, group 1 PH is pulmonary arterial hypertension. It's idiopathic, scleroderma, etc., connective tissue disease, and other types. What about non-group 1 PH? This is a study in group 3, and it's the first glimpse we've had that maybe we can do something for group 3. What is group 3? Group 3 is pulmonary hypertension associated with lung disease. And there are unfortunately many people sick and suffering with lung disease that causes pulmonary hypertension. It's not PAH. It's a very different issue. Well, there was again, an interesting approach, which is to say, okay, we're going to try to give the medications with an inhaler. Why? Because it's going to get down into the lungs, into the areas that are affected the most. And not have side effects to the same degree. And this is a study of inhaled treprostinil in interstitial lung disease. And you can see that it significantly improves 6-minute walk distance. So inhaled treprostinil is effective and available in the United States, but it is not available in Canada. It's never been brought into Canada. And you can see that it didn't really matter if you had a low pulmonary vascular resistance, or, well, it's abnormal, but lowish versus highish versus even higher, okay? Because in all cases, the treprostinil helped reduce the pulmonary vascular resistance. So we're going to call that a success. It would be nice to have access for selected patients, but it's really not available, unfortunately. Now, what about other things? Okay, COPD, chronic obstructive pulmonary disease, huge problem, thousands and thousands of patients, and there's a very large registry called the Go Deep Registry, which has been coordinated out of Germany and Europe, and they're collecting data on thousands of patients, and so they begin to see signals for things that you can't see in small studies. Look at the numbers down here, 5,000, 2,000 patients. And what they showed was that if you have COPD with pulmonary hypertension, your prognosis is worse than even if you have idiopathic PAH. And so what they did then is they said, okay, what about patients that were given pretty loosely, actually, but given sildenafil and tadalafil, those phosphodiesterase inhibitors. And it turned out that if COPD patients with pulmonary hypertension were given a phosphodiesterase inhibitor, their prognosis was improved. It's still not fantastic by any stretch of the imagination, but it does seem to offer some benefit. So we'll have to see moving forward if this pans out in other studies, but at least there was a signal for it. Okay, now I said before that now pulmonary hypertension is defined as a mean pulmonary artery pressure over 20 milliliters of mercury. We've talked about group 1, which is PAH. We've talked about group 3, which is lung disease. Now we're going to talk about group 2, which is left heart disease, which is a huge problem in North America and elsewhere. Because the blood goes in the lungs, but if it can't get out of the lungs into a normal left heart, then the pressure rises in the lungs. And there's a particular group called combined pre and post capillary pulmonary hypertension, where the lung arteries don't like having a back pressure from the left heart, and they react and they narrow even more. And so this is a schema. It's actually a beautiful old study from 1956 looking at people with rheumatic heart disease, which we don't see much in Canada anymore. But imagine that there was a problem in the left side of the heart. Remember, the blood's moving through like this, okay? And so the left heart's suddenly not well, and there's a backup of pressure into the lungs. These are the lung capillaries. And here's your right ventricle trying to pump, okay? And in many people, what happens is not only is there back pressure, but the arteries don't like it and they thicken, which then makes a really high pressure here. And that's what we call combined pre and post capillary pulmonary hypertension. And until now, there's been nothing to do except trying to lower the pressure on this side, which is, first of all, very difficult to do. And second of all, doesn't help that much. You have to get it really low, and that's very hard to do. So people really get a lot of bad right heart failure from the problem on the left side, CP-CPH, okay? And there was nothing to do. Well, there's just been a very recent announcement, and this is all I know about it, okay? Using sotatercept in this group, CP-CPH. It was a phase 2 study, and it met its primary endpoint, which was, again, a reduction in pulmonary vascular resistance. So this is the first time we've been able to demonstrate, and very interesting with an anti-proliferative agent, that we can help the right heart in people with CP-CPH and a stiff left heart, okay? So this is very encouraging. We don't know anything more. This is, hot off the press, so to speak. We don't know anything more, and we'll have to see how this develops. But it would be a major breakthrough if it's is proven in a phase 3 trial, and there are many people who might benefit from it. Okay, to conclude, it's fun to have fun, but you have to know how. This is how we conduct ourselves in the studies. It's very important to move forward. If we don't move forward, we have nothing new to offer our patients, but we have to know how. And so there are a series of rules that I like. Success in a rat, because of course these medications are all tested in animals before, and usually the models are rat models. Success in a rat does not mean success in a human. Of those studies that I showed you that failed, every one of those studies had worked in a rat model. But there's a corollary. Failure in a rat likely means failure in a human. So there are many compounds that are tested that never make it past the rat stage. And so far, that's sort of held true. The second thing we need to look for is we need to think a bit differently. We look at pulmonary vascular resistance, and of course, it's good to reduce the resistance in the pipes, but there are many ways to reduce several ways to reduce the resistance. We are dealing with a disorder that raises the blood pressure in the lungs. And in my view, a medication for high blood pressure should reduce the blood pressure. People take medications for high blood pressure in their body, as what we measure with that cuff in the arm. There isn't a single high blood pressure medication approved by Health Canada that doesn't lower the blood pressure. Many of our previous PAH medications had minimal effect on the blood pressure. They improved flow through the lung, and believe me, they were beneficial in that way, but they really weren't getting to the fundamental issue. I think we need to hold medications to a higher standard now, and we should be looking at all of them as to how they reduce the blood pressure. I must say that the sotatercept studies suggest that sotatercept is more effective at reducing the blood pressure than some of our previous medications. Clinical trials take time and are costly. Patient safety is paramount. Effectiveness really isn't proven until phase 3 trials. And remember that those phase 3 trials pick a very tightly controlled, highly characterized group, and then may well not apply to larger populations, although usually they do to some degree. And we cannot do a single study without PAH patients and other pulmonary hypertension patients volunteering and offering to be part of these trials. It's central to new medication discovery. There are also new improvements in trial design. It used to be, we would design a trial and go the whole way through. Now, there are ways to design the trial such that if an early analysis of the results before the whole study's finished show that one of the treatment arms, let's say a higher dose or let's say a more frequent dose, is offering a lot more benefit than other arms, we can do what's called an adaptive trial and change the structure of the trial to then concentrate on that sort of successful arm. And the other thing I think that's going to be very important to look for in the future is the advent of combination anti-proliferative therapy. I mentioned sotatercept. I mentioned seralutinib. There are going to be others coming along. The question is, how will they work with each other? And will that provide more benefit than a single agent alone? We don't have those answers yet. And with that, I'm going to stop and open up to questions. Thank you very much. Let me stop sharing. Here we go.
Goodness, thank you. Thank you so much for all of that. There's a couple of questions. I'll start off with Miriam's question. She said the end point for anastrozole, and that one, is only 3 months. Is that really enough time to see effects?
Very, very good question. We learned from the trials of sotatercept, which lasted actually 6 months. And again, that was sort of a calculated gamble. I remember the discussions and sort of planning the trial. Would 6 months be enough to show benefit in anti-proliferative drug? Would 6 months be enough to sort of unblock the drain? And so one of the questions, a very good question, is did we go on long enough with anastrozole? And because the study's sort of negative, maybe we didn't. And so we don't know. We don't know. And so again, one of the questions to answer is, okay, let's try to go on longer. But then we need a cohort of patients. We have a limited number of PAH patients, and we have to choose our therapies to try to find the ones that seem to hold the most promise. And remember that they're on top of other therapies. Jamie just put something in the chat, and I think it's very important for people to realize. I'm not talking about using sotatercept alone. Sotatercept is used on top of all of our current therapies. It's never been tested alone. And it could be that I talk about combination therapies. Maybe we do need the other therapies with sotatercept. We don't know yet. We don't. All we know is that we use them all together right now. So yes, could be a study of 1 year versus 3 months might show more effect, but it means committing patients to a 1 year study where in the end the study might be negative. And you then have to ask yourself, okay, how important is the question I'm trying to answer? And how likely is it that I'm going to see a very important dramatic advance by doing it? We're careful with our patients. We're parsimonious with our patients. We try, and when we do studies, try to at least try to answer a question that we feel will be very important in terms of an advance in therapy.
I'm sure it would be hard to get patients to agree to a trial if there was not that promise of substantial benefit.
May I answer the interloper from UK that just put up a chat?
Go ahead.
Okay. A brilliant question, and we're struggling with it right now, and sort of everybody's talking about it. The sotatercept trial did include patients with what I would call simple congenital heart disease, which had been closed and repaired for at least a year and had residual pulmonary hypertension. So mostly atrial septal defects, things like that. There's a whole population of an unmet need, which is people with who've developed PAH because of complex congenital heart disease that's unrepaired. None of these agents, well, except for bosentan, have really been tested in what we call Eisenmenger syndrome. And my dream, and again, I'm putting this out, and it's a hypothesis, okay? Might be that if we get proper antiproliferative therapy, we could take an Eisenmeger's patient, bring them back to a state where they might then be repairable, and then continue the pulmonary hypertension therapy if there's residual PH. So that's a hypothesis. It would have to be tested in trials. And I, there are a lot of adult congenital heart centers that are talking about that. I'd love everyone to be able to put their heads together and make a large international trial, because you need those kind of numbers, and then proceed with a study like that, because certainly those poor people do need some sort of therapy, effective therapy.
Thank you. Okay,
You're
next
welcome.
question, also from Hannah. Um, are there any naturally occurring foods, supplements, etc., that have a similar effect as the inhibitors?
Good question. To our knowledge, to my knowledge, no. Okay. The agents we're using are, if you realize, they're so specific. They're going after one or two or three or four receptors in the entire body. And so we need to attack the disease in a way, receptor by receptor. We need to try and make our best guess as to which receptors are normal. And when I say guess, I mean highly educated guess. There are people working on the cell biology underlying the what all these receptors do in health and disease. We have prominent groups in Canada, and particularly Quebec City and elsewhere, that are working on this, and so they're going to lead us to say, okay, this is a potentially very important target. It's worth going after. But in terms of natural substances, they tend to have very broad effects on things, and so um, uh, I don't know that they would be particularly effective unless again you find a natural substance that attacks one particular or two particular receptors.
Okay, thank you. Next question is from Ian Gibson, who asks, what about other strategies like the modified stem approach in the Sapphire trial?
Stem cells, this was a study led by Duncan Stewart in Ottawa. It was a brilliant study and a brilliant concept. Stem cell therapy in and of itself is very interesting. It's imagine what you're doing is if you believe that well, it's you don't believe it, the science that the in a way the drain's being choked by weeds, okay? And if you could pour healthy cells, nice new grass cells in there. You want to kill the weeds, okay? But you need to reseed, okay? And if you can reseed with healthy cells that are producing beneficial molecules, maybe you can impede the growth of the weeds, okay? And that was in a way the entire concept behind it. And the initial results were encouraging. It went on to another trial, but the trial really got sort of killed during COVID, unfortunately. So the concept's still there. Concept's still there. It just remains to be tested.
Okay, now there's somebody with their hand up. Frank, do you have a question you'd like to ask? Can you put it in the chat or the Q&A? Oh, and I think that's Kim has her hand up. Kim, go ahead.
Sorry, Robin. No, I don't. I was just testing to see if
Okay.
Frank's hand was stuck up.
Oh, Okay. And is it? I think Frank has put his hand down, and I'm not seeing any more questions. Any other questions for Dr. Langleman? We've got a couple minutes left. Um, I had one actually that sort of came up when you were talking about serotonin. I know that didn't succeed, but since that SSRIs are used for mental health, was there any effect on the mental health of people during the serotonin trials?
No,
No.
not really. But you raise an interesting question because there was there have been studies looking whether SSRIs cause PH,
Hmm.
and um, they've been equivocal in adults. There was a suggestion that pregnant women taking SSRIs, their babies had more perinatal pulmonary hypertension in the babies. There was a slightly higher incidence. So serotonin may be very important early on, but the pulmonary hypertension in newborns is a different kind of pulmonary hypertension. It involves very much vascular tone. And so serotonin may be important in the types of pulmonary hypertension that have vascular tone versus plugging inside the vessel,
Hmm.
which is what we see in the adults more. But that again remains to be determined.
Interesting. Okay. Oh, now there's questions coming in. Okay. Uh, Another one from Miriam. Is there any more research looking into the causes of high estrogen and its link to PAH, like hormonal birth controls?
Yeah. So there's a lot of interest in the hormonal effects in pH. The problem is it's very hard to do what we call, clean studies in humans. You can control things very well in an animal study. It's much harder to control things in a human population. And uh, but what is interesting is that many contraception is of course encouraged in pH. Pregnancy can be extremely dangerous in pulmonary hypertension, and so we encourage women and men, I mean couples, to absolutely use contraception of some sort. And again, there's been no real signal that taking hormonal oral contraceptive pills affects pH positively or negatively. Now, the higher dose estrogens had a prothrombotic effect. In other words, they used to predispose to clot, which of course, pulmonary emboli, clots can be dangerous in pulmonary hypertension. But the current oral contraceptives use low dose estrogen, and again, there's no evidence that they either promote or slow pulmonary hypertension.
Okay. Um, Barb asks, is sotatercept used with Adcirca and ambrisentan?
Sure, sure. In fact, all the trials of sotatercept, the people were on at least two background agents, most commonly a phosphodiesterase inhibitor and an endothelin antagonist. So ambrisentan's an endothelin antagonist, and tadalafil or sildenafil is a phosphodiesterase inhibitor. And so pretty much everyone was on that in the studies.
Okay, okay.
Some were on triple agent with a prostanoid.
Okay, there's another one that's sort of similar. For CTPH, can we combine Adempas with another medication?
So right now in CTEPH is a disorder where people, it's about 3% of the population of people who've had a pulmonary embolism, where a clot's generally traveled up from the leg and gone into the lung. Most pulmonary emboli dissolve by themselves. About 3% of people, they stick around and they don't dissolve. And so they become this sort of rubbery, fibrous stuff that sits in the lung and occludes the circulation. Um, the only medication really that's been shown to help patients with inoperable disease or uh, with residual pulmonary hypertension post-surgery is riociguat, which is Adempas, which is that soluble guanylate cyclase stimulator I mentioned earlier. Um, and so, yes, it's approved for use in CTEPH. Now, there are other modalities for CTEPH. There's surgery, which is pulmonary thromboendarterectomy, and increasingly, many patients are candidates for what we call balloon pulmonary angioplasty, which wasn't available a decade ago, at least widely available. And you can help them by doing serial balloons on multiple arteries. In fact, today, before this talk, we were doing a BPA on a patient. So um, uh, the place of the surgery, the balloon, the medication is shifting and drifting, and many people are receiving what we call hybrid therapy, which is they may have surgery, then they may get cleaned up with balloons, and they may be on riociguat to help them. So there are all sorts of combinations you can do to the patient's benefit to help them.
Okay, hopefully we'll get through the last few fairly quickly. Jazz asks, has sotatercept ever been tried in CTEPH patients?
No, has not.
Okay, that was quick. Ian asks, is there one or more causes of pulmonary hypertension, or is it just proliferative signaling problem?
Well, the causes, there's a list of it depends on the groups, but the causes are there are multiple causes. There are probably, 25 identified causes of PH. If we're looking at PAH, we know that they fall into 4 or 5 subgroups. Idiopathic, the patients with genetic variants that cause it, connective tissue disease, liver disease, HIV, drugs and toxins, and congenital cardiac shunts. Okay, so um, uh, it's been tested now in group 1, which is some of those patients. Um, Now we have this suggestion that maybe it does something in group 2, a particular subset of group 2, which is left heart disease. But we don't know more about it yet.
Okay, last question, and this was a good one to end on, I think, from Jamie. What are you most excited for in the next 5 years?
I think we're finally coming around to uh, realizing that cell proliferation is central and that by attacking it, we can finally begin to sort of unblock the clogs. We're seeing some evidence of that with sotatercept, and we need to push harder and with other agents as well. And that's incredibly encouraging. When you look at those prognostic curves, they're dramatically better.
I think that's an excellent note to end on. So I will stop the recording there. And thank you so much, Dr.
Thank you
Langloben,
for
for all
inviting me.
your presentation and your answering questions. You always explain things so clearly, and it's just so wonderful to have you. So thank you,
It's
and thank you, everybody, for coming.
always a pleasure. Thank you so much for attending. Thank you.